I would now like to welcome Brendan Cordeiro, who will be joining from St. Michael's Hospital, the Keenan Research Center. And he will be discussing sex differences in the effect of obesity on adaptive immunity in a model of multiple sclerosis. So I will let you take it away. We can now see your screen.
Perfect. Awesome. I just want to thank the organizers for giving me the chance to show my work here. So as mentioned, I'm a postdoc in the Dunn Lab over in Toronto. So my project is sex differences in the effect of obesity on adaptive immunity in a model of multiple sclerosis. So our lab focuses on multiple sclerosis, or MS, which is a chronic autoimmune disease of the central nervous system. So MS affects over 2.5 million people worldwide, with Canada having the highest prevalence of the disease. So in MS, inflammation in the CNS damages the myelin sheath that insulates neurons, and over time this damage accumulates and leads to progressive disability and death. So what causes the initial autoimmune reaction in MS is unknown, but both genetic and environmental factors are thought to be involved. So one such factor is female physiology, which increases MS risk by more than threefold, as one of the largest risk factors for developing this disease. What's interesting is that the ratio of men and women diagnosed with MS has been steadily increasing over the past 5 decades, and is now close to 3 to 1 females for every male in most developed countries. So this suggests that an environmental factor is interacting with female sex to increase MS risk. One such environmental factor could be adolescent obesity. So over the past 40 years, the worldwide prevalence of obesity has been increasing in all age groups, and this includes over 120 million children and adolescents. Now, obesity is known to induce a chronic low-grade systemic inflammation and is also known to promote the acquisition of autoimmune diseases later in life. Now, multiple epidemiological studies have identified that adolescent obesity is strongly associated with the development of MS later in life, and several of these studies found that this association was significant only in females. So this led to our overarching hypothesis that female sex and obesity are interacting to enhance CNS autoimmunity. So to study this, we made use of the diet-induced obesity model, or DIO. It's a very simple model where you take your favorite mouse strain, such as C57 black 6 mice, and feed them either a normal child diet or a diet that's high in fat, and this will promote adiposity in the mice. Now, historically, this model has mostly been used in male mice. That's because male mice are very prone to the weight gain from this diet and also acquire a type 2 diabetes-like metabolic syndrome that closely mirrors what goes on in humans in obesity. Female mice, on the other hand, tend to be resistant to the metabolic syndrome acquisition. So the first thing we had to do in this project was try to find a time course of high-fat diet feeding that would lead to significant weight gain in both sexes with very minor metabolic disturbances in the male mice. So throughout my presentation, the blue data points represent male mice, red represents the female mice, the closed circles are the high-fat diet-fed mice, and the open circles are the normal child diet-fed mice. So here we did a time course of high-fat diet feeding, and we found that after 4 weeks of high-fat diet feeding, we started to see a significant weight gain in both sexes of the mice. And then we measured fasting glucose levels in the serum, and we found no significant change. However, when we ran a glucose tolerance test, we did see that the male mice on high-fat diet had a bit of hyperglycemia, but when we ran an insulin tolerance test, we saw there was no significant change in insulin resistance. So therefore, 4 weeks of high-fat diet is what we went with because we had increases in weight with only minor metabolic disturbance. And so next we want to induce MS-like disease in our mice. So to do this, we used our model, the experimental autoimmune encephalomyelitis, or EAE model. So simply you inject mice with myelin oligodendrocyte peptide in an emulsion with complete Frin's adjuvant along with 2 doses of pertussis toxin. And this induces a MS-like disease in the mice, which is characterized by an ascending paralysis that can be scored using a clinical score chart. So here's the typical EAE response curve. And at endpoint, we can basically get an idea of how severe EAE disease is in the mice. So when we induced EAE disease in our mice that are fed a high-fat diet, we found that in both sexes, high-fat diet promoted disease severity. But this was especially true in the female mice. So if you look at cumulative scoring, as well as the score at the peak of the disease, we saw the disease was significantly more severe in the female mice fed a high-fat diet. And if you ran histology to look at the percentage of demyelination in the CNS of these mice, we found that the female mice had a significant increase in demyelination compared to normal child diet. So this suggests that the 4 weeks of high-fat diet is promoting EAE disease severity. So of course, next thing we want to check is what's going on with the immune system in the CNS of these mice. So again, we induce EAE in the mice, but this time at the peak of disease, we would then harvest the CNS and characterize the T cell compartment via flow cytometry by looking at pro-inflammatory cytokine production as well as activation markers. And what we found, just looking at the total number and frequency of CD4 T cells in the CNS, that in both cases, male and female mice fed a high-fat diet have a significant increase in the total number of CD4 T cells. When we then look at the pro-inflammatory cytokine expression of these CD4 T cells, what we found is that in the female mice fed a high-fat diet, there's this increase in total number and frequency of these double positive IL-17 interferon gamma producing cells, as well as GM-CSF interferon gamma producing cells. So these double positive cell subtypes are known to promote severe EAE disease, as well as promote severe MS disease in human patients. So this suggests that in the female mice fed a high-fat diet, there's this large increase in the pro-inflammatory response. So next we want to look at, okay, what's going on with the myelin-specific T cells. So same idea, except this time we look at the early phase of the disease where most of the MOG-specific T cells are present in the spleen. And we can characterize these cells by flow cytometry by using a MOG-specific tetramer to gate on the myelin-specific T cells. And we can look at their expression of activation markers by flow, as well as re-stimulating the cells with MOG peptide to look at their pro-inflammatory cytokine production by ELISA. So what we found is that in both sexes, feeding the mice a high-fat diet promotes the frequency of MOG-specific CD4 T cells. But if we look at the total numbers, it's only increased significantly in the female mice fed a high-fat diet. If you re-stimulate the cells with MOG, what we found is that in both cases, high-fat diet promotes the production of interferon gamma, but this is especially true in the female mice. If you look at IL-17, on the other hand, the male mice fed a high-fat diet have a significant production of IL-17. Compared to the normal child diet. So it seems like in the female mice on high-fat diet, there's this promotion of this Th1 CD4 T cell response. So we want to see, okay, is this true without EAE? How about in a naive CV7 Black 6 mouse? So we fed normal mice, naive mice, a normal child diet or high-fat diet, and we found that in the female mice, a high-fat diet promotes the increased frequency, a significant increase in the frequency of CD4 T cells. If you drill down on these T cells and look at the expression of CD44, we found that there was a significant increase in the frequency of these CD44 high memory CD4 T cells. And also these T cells have a significantly increased CD44 expression, suggesting that they're more activated. If you re-stimulate these T cells with CD3CD28, we again see this promotion of an interferon gamma production in the female mice fed a high-fat diet. So again, we're seeing just generally in the mice without EAE, the skewing of the immune response towards the Th1 due to the high-fat diet. Now, what exactly is causing this? So we first want to see if the weight gain is important for this phenotype. So to test this out, we gave our mice high-fat diet, but then we transitioned them back into normal child diet, essentially putting the mice on a diet. So what we found is that the percentage of weight difference is decreased in the mice when they're put back on the normal child diet from being on the high-fat diet. And if we look at the T cell compartment, we again see a reduction in our frequency of CD44 cells in the mice that are put on the diet. And if we re-stimulate the cells of CD3CD28, we lose the increase in interferon gamma production. So it seems like the weight gain is important for this phenotype. But what if the diet itself is pro-inflammatory? So to test this, we used SJL mice, which are resistant to gaining weight on the high-fat diet model. So when you feed these mice a high-fat diet, we see no significant change in weight gain, which is consistent with the literature. And we also don't see any change in the frequency of CD44 cells. And we also see no change in the production of interferon gamma when we induce EAE in these mice using the SJL equivalent to MOG, which is PLP peptide. So you re-stimulate the T cells with PLP, we see no difference in interferon gamma production. So it seems like the weight gain itself was important, not so much the diet itself. So the diet is not pro-inflammatory in this case. We next want to see, okay, so what about ovarian hormones? So we either performed a sham surgery or an ovarectomy on our mice, thus removing the ovaries, removing the production of ovarian hormones. And what we found is that if you take the T cells from these mice and re-stimulate them with CD3CD28, that the mice that received an ovarectomy that were fed a high-fat diet have a significant reduction in interferon gamma production compared to the sham surgery mice on a high-fat diet. So this suggests that the diet itself, the weight gain is important, and this is interacting with ovarian hormones. So how are these two factors interacting to promote this Th1 response? So there's a lot of data I'm not going to show here. We showed that it's not anything to do with the antigen presenting cells. We also show there's no effect on the Tregs. Instead, we found that intrinsically in the CD4 T cells themselves, there's this promotion of Th1 associated gene expression. So a lot of STAT1, TBET, interferon gamma gene expression. So intrinsically, something is acting specifically on the CD4 T cells to induce this Th1 response. Now, one factor that's known to promote Th1 skewing in CD4 T cells as well as other immune cell types is interferon alpha. So interferon alpha is a type 1 interferon, signals through the interferon alpha receptor on CD4 T cells. And we found that in the female mice fed a high-fat diet, if you look in the serum, there's a significant increase in interferon alpha only in these mice. So to test whether or not the interferon alpha signaling in the CD4 T cells was responsible for this phenotype, we obtained interferon alpha flux LCK Cree mice from the lab of Dr. David Brooks. And so these mice have no interferon alpha receptor expression specifically on T cells. And we found that looking at the weight change, if we give these mice a high-fat diet, there's no difference in the weight change. But if you look at our frequency of CD44 high cells, there's a significant reduction when you remove the interferon alpha receptor from these CD4 T cells. And similarly, if you restimulate these cells with MOG, we find that the interferon gamma production is significantly reduced in the mice that are on high-fat diet with the interferon alpha receptor removed from the CD4 T cells. So finally, what does this mean for EAE disease severity? Is interferon alpha responsible for that? So we induced EAE disease in our wild type or interferon alpha receptor flux mice. And what we found is that when you feed the mice a normal child diet, there was no significant difference in EAE disease severity. But as soon as you gave the mice a high-fat diet, not only was the EAE severity reduced in the interferon alpha receptor flux mice, but there was also a delay of onset of disease. So in conclusion, on a normal child diet, female mice have a regular amount of interferon alpha in the serum. This leads to normal Th1 signaling and a normal amount of interferon gamma production. But on a high-fat diet, specifically in the female mice, there's this overabundance of interferon alpha that signals through the interferon alpha receptor, leading to a robust promotion of the Th1 signaling cascade, more interferon gamma production, and more EAE disease severity. So I'd just like to, of course, acknowledge my lab, of course, my supervisor, Dr. Shannon Dunn, as well as members of my lab, our various collaborators, and of course, the funding. Thank you very much for your attention.
Thank you very much, Brendan, for your presentation. And I will ask a quick question, but if anybody else has any questions, please put them in the Q&A section. So I would like to ask, so you focused on ovarian hormones, and I was wondering if there are any specific ones that you're interested in and that you may believe are causing these changes, and if you think any differences in higher or lower estrogen or progesterone levels might actually cause this.
So we're very interested in estrogen. Estrogen is known to promote Th1 signaling in T cells. So estrogen, there's an estrogen promoter site, binding site on the promoter of interferon gamma, for example. So that's kind of the one we were really focusing on. But also, there's a lot of recent work showing that progesterone also plays a huge role in the immune system. So future work, we're going to look at both. So that's kind of, we just did the ovarectomy as just like a general way of looking at it for now, but we plan to expand on this in the future.
Great. Thank you so much for answering my question. And I would like to direct you to just answer via the Q&A section. So type in your responses because we now need to move on to the next presenter. But thank you so much for your amazing talk.
Thank you.